Yeah, so a mutation is just a tiny error in our DNA, and it's responsible for pretty much all the genetic diseases that we know. Genetic diseases are caused by genetic mutations that are um, just errors in DNA. So something is wrong. And since DNA is so important, because DNA is in every single one of our cells, a tiny, tiny mistake in the DNA can cause really big problems. But sometimes, in very rare cases, they can even be good for us. Mutations can be good for us, even though most of the time they cause diseases. This is because, let's assume you have a really big book, right? A really important book. So let's say, I don't know, the Bible, okay? Let's say you have the Bible, and you can, let's say you have somebody who's copying the Bible word for word. So they're just reading through the Bible and they're copying everything word for word. And if they mess up one word in the Bible, it could be a really big error, right? It could be telling people to do something bad instead of something good. Except in a rare case, if somebody makes an error while copying the Bible, what if they make a good change? What if they change something to make it even better? I don't know. Maybe I shouldn't have picked the Bible for that example. But like I said, DNA is really important, right? If you make a tiny mistake while copying it, it can be a happy mistake sometimes. Like you can have a mutation, which is a change in your DNA, a random change that causes you to have even better vision, for example. Or you could have an error in your DNA, which is much more likely that's going to make you blind permanently. So this is why an error in a DNA in your in your DNA sequence could be in rare cases it could be really good, but most of the time it can be really bad. So when a gene is mutated, like when you have a copy of a gene that's yeah. So and then natural selection happens. So usually what happens is if you have a mutated gene that's actually good for you, like you end up having really good vision, then a special process of evolution is going to occur called natural selection, which we might talk about later, which is going to cause that gene to become more frequent in the population. So that's pretty much how evolution happened. That's how like monkeys evolved into humans. Not exactly monkeys, but something like monkeys evolved into humans because of all these little random changes in our DNA that made us better and better. So a mutation in somewhere in our DNA caused us to become smarter than monkeys, for example. And that's a really random change, and all these random changes combined um, are responsible for all the differences between species. They're all tiny random changes that piled up over time. And
But
Ishan,
if a
so
monkey,
oh sorry,
if
yeah, what?
if a monkey becomes blind, later will humans become like totally blind?
Oh wait, sorry, what did you say? Like, could you elaborate on
Is
that?
it only is it only the good stuff that allows us to evolve, or can like bad stuff be like? Can bad stuff also allow us to evolve?
Oh wow,
Like, what
that's
if we like?
yeah, I know what you mean.
What if monkeys became blind?
Okay,
What if
see,
monkeys became
that's
blind?
an amazing question. That's pretty deep. So usually we think of evolution as a bunch of good things that piled up. Okay, we think of evolution as all these changes that made us better and better and better over time, and eventually we got so better that we're where we are today, and we're better than monkeys. So it's well, it's not exactly like you said about like monkeys becoming blind and that evolving, but you can have things that give you a benefit, okay? So essentially, the way natural selection works is if you have something that makes you better and more able to survive, then um like let's not consider humans. Let's consider like monkeys, okay? So if a monkey is really smart and they're able to make tools and they're able to survive, then that means that they're going to live longer and they're going to have more children, okay? So anything that's able to survive better is going to live longer and it's going to have more children. That's pretty much the way it works in nature. And when you have more children than other monkeys, that means that more of your DNA, because your children carry your DNA, right? They carry your traits. That means that more of your children are going to be present in the population. So population meaning just all the monkeys in that area. If you have a whole jungle of monkeys, if one monkey has more children than the others, then that means that that monkey's DNA is going to be more is going to be is going to have a higher frequency in the population of monkeys in the jungle. And if those monkeys also carry the trait that makes them smarter, makes them able to make tools and able to live longer and have more children, then that means that it's gonna grow exponentially. It's gonna grow more and more in the population. So that's the way it works when you have a good mutation, something that makes you able to live longer or and have more children. Because the only reason that anything becomes more frequent in the population is because it allows you to live longer. Is it yeah, it allows you to live longer and have more children. It's the same reason why like some animal develop the ability to swim because it can swim away from predators, so it can avoid danger, so it can live longer and have more children. That's the only reason. Um, so since it has more children, that trait becomes more frequent in the population, and as eventually, like most of the species of monkeys in the world might have that trait. So because it becomes so frequent, because it's so good for monkeys and helps them survive and have more children, it just takes over the whole world. And sometimes, okay, I'll get to the eye color questions in a second. So yeah, sometimes your mutations can't be entirely good for you, but they can also help you survive in some way. So they end up becoming more common. And we'll get to that. That's a pretty good transition into our next thing, which is called sickle cell anemia. It's a special disease, okay? So sickle cell anemia is caused by a tiny mutation. It's a tiny mutation in the DNA. It's just one little letter that's wrong. And this causes your red blood cells, the blood cells that carry oxygen in your body, to have a different shape. And this causes them to sometimes get stuck in very small blood vessels, so you can't get blood to your organs as well. And this usually affects like African American people more than other races. So it's just something that's more frequent in the African American population, and we don't see a reason for why it's frequent, because usually things that are bad for you, things that are bad for any organism in nature, are going to make it die before it can have children. So you would expect it to not be very frequent. Except this thing is way more frequent in the African American population than in other populations. And the reason for that is actually, even though it's a disease, it actually has a hidden benefit, which means that it protects from malaria. So malaria is something that affects red blood cells, and it's a disease carried by mosquitoes. And even though this is technically a disease, it's a special hidden perk that this protects from malaria. And malaria is especially common in Africa. So that's why initially this evolved. It became more frequent in the African population because uh, because this mutation protected them from malaria. So the people who had this mutation, even though sometimes they died because they couldn't get blood to their organs, it could protect them from malaria. So as a whole, it was somewhat beneficial. So people who had it lived longer and they had more children, and that's why it became common. So this is technically something bad. So the person who was asking that question, how can a bad thing evolve and become more frequent? Yeah, this is something bad. It's a disease that doesn't allow you to get blood to your organs. But even though it's bad, it protects from malaria, which is a deadly disease. And that's why it became really common in a population that had to be protected from malaria. So I just went on a long rant there. I hope some of that made sense. And yeah, so just as a little recap to get back on track. So when a gene is mutated, when the sequence changes and it becomes mutated, it's passed down to children, just like a normal gene, even though it has a slightly different sequence. So mutated genes, they can be dangerous. They can they can be deadly diseases, and they're also passed down to children, just like normal genes are passed down to your children. So sickle cell anemia is passed on to children, just like the trait for making normal red blood cells is passed on to children. So that's why sickle cell anemia can be like, if your parents have it, then you likely end up having it. Um, yeah, that's pretty much the reason why genetic diseases are common. They run in families instead of if your parents have it, there's no difference in the likelihood of you having it. So I'll get to some questions in the chat now. Um, So Ishan, so if someone has one blue eye and one brown eye, is that a mutation? So pretty much every part of your DNA is because almost every part of your DNA, like any interesting changes, are because of a mutation. Like for example, a long time in the past, somebody had a mutation, a little random change in their DNA that caused them to have blue eyes instead of brown eyes. And that trait just became passed on, like it got passed on to their children, and that became really popular, really common in the population. So this is this is a case of mutation because somebody has a special quirk in their DNA, a random little change in their DNA that helps them have one blue eye and one brown eye. It doesn't really help you. It just does nothing, but it's not dangerous. So it's not really a factor in like evolution or anything. So yeah, it's a mutation that causes you to have an interesting trait. Arpon, if your mom had blue eyes and your dad had green eyes, can the kid's eye be aqua? So I can't answer this question like for sure, like in terms of the facts. But what I can tell you is that there are multiple genes controlling eye color. So it's possible that if your mom has blue eyes and your dad has green eyes, you can be somewhere in between. You don't have to have one or the other. So because there's multiple genes controlling eye color, you can be somewhere in the middle. But if there were only one gene controlling eye color, you would have to have only one trait or the other trait. So yeah, it's technically possible for the kid's eye to be aqua, I would guess. But it's in a case where it's only one gene, you can't have a combination of two traits really in most commonly. So Arpon, yeah, someone with sickle cell can definitely get a heart attack or a stroke if it closes those arteries. Like the coronary artery, for example, that's the artery that's usually blocked when you have a heart attack. Your heart muscle dies because it doesn't get blood. So sickle cell is really dangerous for people who do a lot of exercise, because when they have exercise, they need a lot of blood. And these sickle-shaped red blood cells, they can get caught in the middle of these tiny blood vessels, which causes them to not have blood. Like it can cause a stroke or a heart attack, and it can lead to death. Um, so yeah, just to collect our thoughts a little, I talked for a long time there. Um, so the person who was asking if evolution can be it can spread bad traits. Did that sort of answer your question? Are you still on the call?
Yes.
Okay, yeah. So did that make sense a little? The only reason that a trait becomes more
It made
common, huh? Sorry, what?
it made sense.
All right, awesome. Yeah. So yeah, thanks for that question. That took us down a little road where you can, yeah. So you can evolution really picks stuff that is better for people as a whole, but it can still have cons to it. Like sickle-shaped red blood cells, they can cause a disease that kills you, but at the same time they protect from malaria, which made them more common in the African American population. So this is just a little link. I guess I'll post it in the chat, but it's not really that long. It's just a little link talking about different kinds of mutations, and you can I'll screen share and I'll go through it, but you can just follow along if you would like to. So this is the first interactive little thing we're doing. So this is an example of how different genetic mutations can cause different diseases. So this is the process of development from like the parents to like the different cells that multiply and make up a kid who grows up to be an adult. So yeah, so you can just click some of these things on the side and you can see the different kinds of mutations that cause different diseases. So when a mutation is inherited from a parent by offspring, so if you remember inheritance, that's just the thing we talked about earlier, where it's how parents pass on their genes to children. So if a parent has a mutated gene, a mutated version of the gene, then this means that if they have kids, the kids will also carry this mutation. So all the cells in the kid are going to have the same mutated gene that the parent has, given that it's passed on to the kid. So that means that this kid, they also have this mutated gene in all their cells. So when this kid has kids of their own, those kids are also going to get the mutated gene. So an inherited mutation is just any mutation that's passed on from parents to kids in such a way that the kids, all the kids' cells have that mutation. So inherited mutations include not only sickle cell anemia, but there's also other diseases called cystic fibrosis, for example. And cystic fibrosis is just a gene. It's just an error in a gene that creates. It has to do with mucus production. So people really have trouble breathing. It's just another example of a genetic disease. And you also have something called Huntington's disease. So Huntington's disease is something that it's also an inherited disease. So if your parents have it, you have a really high likelihood of having it. And this usually happens like when you're an adult, like in your 40s or something. Usually you just have like a bunch of troubles with like moving your muscles and everything. And there's also like you can't think as well, like mental decline. So you can also have mutations that happen while you're an embryo, like your own cells while you're an embryo are messed up. So some cases of autism, so like where you have errors with your brain development or something like that, like there's errors in your brain. This happens sometimes in the embryo, when the embryo itself, like when you're very early in your development, if you have a little mutation like this can cause autism. And oh, we are not going to cover this thing. Okay, so now another really important case of mutations is when your own body cells as an adult, like not when you're very early in your development, not in your parents, but in you get a new mutation that messes up the DNA in some of your cells. So usually what this leads to is cancer. So you definitely heard of cancer before, right, guys? So cancer is like something very dangerous and it happens to a lot of people. The reason cancer happens is because some random DNA changes in some random cells in your body cause the cells to divide uncontrollably. So usually your cells are always like keeping in line, they're following orders, but cancer is a change in the DNA that instructs the cells to keep dividing, keep on dividing and dividing until they form something dangerous called a tumor, which is just, a clump of cells. And that tumor just grows and grows and can break off and get into other parts of your body. Essentially, it's just like it steals resources from your body. Um, so cancer is just uncontrolled cell growth. If you see in the definition here, cells that divide uncontrollably. That's just caused by mutations in your DNA. And these mutations can also be sort of inherited. They're not the mutations themselves, but you can have a higher likelihood of having a certain form of cancer if your parents have it. It's something like that. So, since these mutations are so important for like your genetic makeup, and your likelihood of having different diseases, oftentimes a person can go to a doctor and test if they have a genetic mutation. Like if you have like if you want your son to play sports, you would want to know if he has a genetic disease that could, get him into serious health problems if he plays sports. Like he could get a stroke or something. So, for example, for sickle cell anemia, like exercise can cause the blood vessels to get clogged up, right? So you want to test for a genetic disease as early as possible so you can see if you have it and you can act accordingly. So if you go to a doctor and you want to see if you have a genetic disease, the first thing is that the doctor has to extract your DNA. We're going to do a little lab on that later, but extracting DNA just means that you take a sample of the person's cells, because like we said, every cell in your body has the same DNA. Every cell, like your skin cells, your cheek cells, your nose cells, whatever. All of them have the same DNA. So you just take a sample of any cells in the human body, and then you do some special chemistry to isolate the DNA, meaning you keep only the DNA and you get rid of everything else. And then once you have the DNA, which is isolated, you have only the DNA and none of the other stuff that cells have in a test tube, then you can do a bunch of interesting tests on it. You can sequence the DNA because DNA is a really long molecule that has a bunch of letters lined up. You can think of them as letters. And that's pretty much the way that DNA codes for information. So there's special machines that doctors can use to test the DNA sequence and see if there are any known mutations. So if you go back here, this is a very short, like 3-letter thing of DNA, 3-letter sequence. So if you sequence the person's DNA,
Bye.
oh, sorry, someone say bye. Okay, bye guys. See you at the next class, I guess. So if someone has
Can you send the attendance link?
oh, okay, yeah, hold up. Saloni, can you get the attendance link real quick? Yeah, hopefully Saloni's there. All right, I think I'll get it. So yeah, guys, it's a 6 o'clock. If you want to stay and learn a bit more, because we have some interesting labs and stuff that we're gonna do. If you want to stay, you can stay. Okay.
Attendance?
Yeah, I'm sending the link right now. Hold up a second. Sorry. Yeah, if you want to stay, you can stay. If you want to leave, I'm posting the participation link
I
that
already
you can fill out.
posted it.
Oh, you did? Okay. Yeah. So if you gotta go, see you guys in the next session. Yeah. So if you want to stay, it's great to have you here, though. Okay. We'll continue on with the lesson. Yeah. So yeah. So if you see here, there's a tiny letter that's changed in the DNA sequence. So a doctor can usually look at this and they can tell that, oh, this person has this letter in their DNA. They have a mutation, which makes them more likely to have this disease. So that's just an example of what you can do with DNA. And another thing you can do is a process that's called gel electrophoresis, which we'll also go over. And this is just for separating the DNA and seeing the differences in the DNA length and everything. So after testing for the disease, and if they see that, oh, this person has that disease, they can use a bunch of treatments to try and control it, or they can use something called gene therapy, which we'll also go over to try and fix the problem. So I don't think we have the time for this. I'll go over the next thing quickly so we can because this is more important, and we can go back to this at the end if anyone wants to stay. Okay, so we skipped that, and so gel electrophoresis is one of the procedures that doctors use to test if people might have a genetic disease. So gel electrophoresis is just separating DNA fragments by length. Okay, so keep that in mind. And they separate the fragments by length. And then once they separate those, they get a cool little pattern looking thing like this. And using this pattern, they can tell if somebody is more likely to get a genetic disease. So here is the link for this. If you want to follow along, feel free to. You can click that and follow along with the little lab. You will need Flash Player. So if you can't run it, then don't worry. You can just follow along with this thing here that I'm doing on my screen. Okay. So you start off over here, okay, with a plastic tube. So this is just like a really tiny test tube. And in this test tube, you have done a bunch of chemistry to isolate the DNA. So all that's in this test tube is the DNA of the person you're trying to diagnose. Okay, let's say that. So again, this is what DNA looks like. It's a really long molecule, okay, and it's very, very long. Like if it were thick enough, you could see it with your naked eye, like without any microscope or anything. But it's really thin, which is why you can't see it. And it's just going to be in this test tube right here, floating around in a liquid. And the reason that doctors use gel electrophoresis, okay, that's a big word. It's to separate the DNA so you can sort it and organize it and see what's going on. Because you can't really touch it and sort it with your hands, right? So this is, I guess you can pretend you're this person, a scientist, and you're about to use gel electrophoresis. So first, let's go to the first word of that, okay, gel. So the gel that you're using is sort of like a filter. So if you think of a really dense forest, like a forest with so many trees really close together, if you think about a small person, okay, a really skinny person versus a really big person, like a sumo wrestler. So imagine both of you are trying to run through the forest. The sumo wrestler who's bigger is going to have a harder time running through the forest, so they're going to be left behind. Except you, if you're really skinny, you're going to be able to run faster between the trees and everything. So it's a similar principle here. If you think about the gel as a really dense forest with a lot of trees really close together, it's going to be harder to move through this gel if you're a DNA fragment that's very, very long. Except if you're a DNA fragment that's really short and you can move through easily, quickly, then you're going to move farther. So at a microscopic level, that's just pretty much what's happening here. So you have little holes at the end of the gel, which is where you place the DNA samples. And the special thing about DNA that's used in this case is that DNA has a negative charge. So if you don't already know this, opposite charges attract, okay? So since DNA is negative, if it has a negative charge, it's going to be attracted to positive charge. So using an electrical field, you have positive charge at this end, which attracts the DNA over to this end. So that's the reason why DNA moves through this gel. And even though it's moving in this direction, the reason that it's separated by length is because, like we said, the longer DNA strands have a harder time moving through this dense gel. The shorter ones can move farther in this direction. So if you look here really close up, you can see that the DNA is just going to slither through the gel. It's going to be separated out. And you can see the really long ones are moving a shorter distance. The shorter ones, as you keep the electrical field on, they're just going to keep going farther and farther. So this is how they're sorted. You can use the length of the DNA as a reference for what the DNA actually is, like what the fragment has inside it. So even though like you can't see a single DNA strand because it's really thin, right? If you have a large group of DNA strands that are all clumped together and you use a special chemical to make them colorful, then you can see these because they're clumped together, they're really colorful, and you can see them as bands. So this is what they look like. If this is the gel, after you use the electrical field to separate them, you can see that you have these little bands here, and this is just DNA that's clumped up. So if you want to run a gel for gel electrophoresis, actually, no, we're going to skip this. We're running out of time. So that's pretty much the whole concept of it. You run a gel and you separate it by length using an electrical field. And the special thing is that this DNA is chopped up into tiny pieces before you do this, okay? So this DNA is chopped up at very particular places. So if you think about the DNA as a long string or like a long piece of yarn, you're gonna chop this DNA with special chemicals at very particular places, like you're taking a pair of scissors and you're cutting them, okay? You're cutting the yarn at very particular places. So what's different between people is sometimes in these places where the thing cuts, they have a mutation. So you can use this, you can take advantage of this to diagnose different diseases. So you can see if they have a certain change in their DNA sequence that makes the scissors, the chemical scissors, chop at different places. And remember that if you have a different length of DNA, it's going to end up at a different point because the longer ones move a shorter distance. So you're going to basically end up with a unique pattern here because of different lengths in the DNA fragments. So in this case here, you have a particular genetic condition that's being diagnosed by gel electrophoresis. You take these people's DNA, you separate it by length after chopping it up with scissors, and you realize that some people who have this condition have this particular pattern. So a doctor can use this after isolating your DNA. They can use this as a way to see if you have a particular condition. So recently, very smart people are using experimental techniques. So it's really new, and they're trying to develop something to treat these genetic conditions. So usually in the past, what they would do is they'll just like say, "Oh, I'm really sorry you have this condition. There's nothing we can do about it." But now we have something called gene therapy. So what's wrong when you have a genetic condition is a gene, right? So what gene therapy's aim is, it's trying to make healthy genes get expressed inside diseased cells. So if you have a messed up gene that's causing you to be blind, then the goal of this is to try and deliver a gene into your eye cells that lets you see clearly, because it's using a different gene than the one that you have inside your nucleus. So this is really experimental. This has rarely been done on humans, and they're just starting to do it now over the last few years. And we're going to go more over this in a future class. So guys, I think we don't really have time for this video here. Are you adding it or cutting out the bad one and replacing? Oh, yeah, Arpan, that's a really good question. So he asked, when you're using gene therapy, are you adding the gene into the cell, or are you cutting out the bad gene and replacing it? So recently, what they've been doing, so actually I'll go back. A few years ago, what they discovered was if you can add special sequences of DNA into cells, you can inject it. And it's not just DNA, sometimes it's another molecule called RNA, which you can inject into cells. Like if your eye has a problem, you can inject eye cells with a particular sequence of DNA. Yeah. And you can use this to express the gene inside the eye cells. So even though it's not your own cell's genes, it's still going to express it. So it's not fully effective because it's still a foreign DNA sequence, but that's what they've been doing over the past few years. And recently what they've discovered is that you can use a special thing called CRISPR, CRISPR, which is a new technology that we're going to go over in a future meeting all by itself. And you can cut out the bad gene and replace it with a good gene. So that's a really good question. You have the perfect scientist mentality, you know? So recently what they've been doing is they've been cutting out the bad gene with special chemical technologies, and they're replacing it with a good gene. But this is really dangerous because we don't know exactly what you might be deleting and replacing. So yeah, that's what they've been trying to figure out over the last few years. Even up until right now, they're still working on this in hundreds of labs all around the world and trying to figure out how you can delete only the bad gene and replace it with only the good gene. So yeah, and actually, yeah, thanks, Anna. I'm going to share the video link. So this is an interactive video about gene therapy, and you can follow along with it, answer some questions. We don't have the time to go over it right now, but I just posted it in the chat, and you can see that, like once you click on it, you can see that there's a video. It's like 8 minutes, and you can just answer the questions, follow along, and it's really fascinating. And yeah, Anna. So she's asking, even if you delete more than bad genes, won't it be replaced with good genes anyways? So not exactly. What you're doing when you delete a bad gene is you're sending chemical scissors into the body, and you're saying, okay, wherever you see this sequence, which is the bad gene, I want you to replace it with this gene. So let's say you send out a chemical machine that deletes a gene responsible for bad eye vision, I guess, and you want to replace it with a gene that causes good vision. Except the problem is, if it's deleting the wrong sequence, it's going to replace even good sequences in your body with the gene for, I don't know, vision. So you're going to have right genes. Yeah, they're still going to be good genes, but they're going to be in the wrong places. So this is something called off-target effects, because your target is going to be the bad gene, which you want to replace. And if it's off-target effects, what that means is that you're having the right effect, but you're doing it off the actual target that we want to do. And yeah, it's something like you could replace your taste with sight. That's not exactly what happens, but yeah, the DNA responsible for taste might be in the place for the DNA responsible with sight. So
Wait,
that might mess up your...
tongue?
Yeah, what?
You can see with your tongue?
No, not exactly. So you can't see with your tongue, which is the point. So you're just going to end up losing your sight or losing your taste or something like that. Because you can only have the right gene sequence if it's in the right place. You can only express the right gene if it's in the right place, because that's the way your machinery works. So we only have 1 minute left. So I like I said, I posted the video in the chat if you want to watch it and follow along. And yeah, we're gonna record this and everything. So if you missed any information, it's gonna be on our YouTube channel, which again we'll email out to everyone and we'll link it on our website. Okay, in a while. Thanks for coming. We have less than 1 minute in the meeting, so I got to end it now. But y'all have been really good students, asking a lot of good questions, and I hope to see you at the next meeting, okay? If you have any more questions, feel free to email us and I'll answer. Okay, guys? Talk to you later.
Thank you.
Yeah, thanks for coming. Bye.
Thanks. Bye.
Bye.